Thank you for your interest in this talk. My name is Guy Midgley, and I'm a slightly lapsed but recovering plant ecophysiologist. I head up the Global Change Biology Group at Stellenbosch University, and I teach the topic of climate change and global change at Stellenbosch. Alistair and Kath asked me to provide a talk to this forum on how succulent plants function and the relevance of this for the role of succulent plants in Thicket. There have been some interesting discussions around succulents and its role, and even suggestions that species like Portulacaria afra are able to suppress wildfire through their dampness. I have myself witnessed an invasive grass fire in Fynbos run into a patch of Carpobrotus edulis and stopped dead in its tracks. On the other hand, I've seen a wildfire rip through Tyronia paniculata and Russia Caroli dominated felt in the Worcester-Robertson area, basically melting succulents like Tylochodon paniculatus, cotyledon or biculata, Euphorbia mauritanica, and Euphorbia vermani as it goes. But nonetheless, it's an interesting idea, this how succulents might affect fire. But musings aside, I decided to delve into the notion of the rise of succulents, its evolutionary origin and diversification, and try to relate this to the thicket biome. The talk will therefore be quite speculative, and it's really designed to fuel a discussion rather than answer any questions at this point. Succulent taxa seem to have arisen over quite a range of timescales, and thus explain some of the interesting patterns in family-level global distributions, as indicated on this map. Some groups have their origins long enough ago to have multi-continental distributions, Euphorbia being an example. In this map, one can see how the Euphorbiaceae straddle the Atlantic, as do the Crassulaceae, while more recently originated Cactaceae and Azoaceae have more distinct single continent distributions. According to this map, Australia has no succulent hotspots. Euphorbs and aloes have their origins in Africa. But while euphorbs originated early in the tertiary, aloes more likely originated in the Miocene, so much more recently. On the left panel here, we can see how aloe species migrated and radiated across Africa from a putated southern origin, even crossing to Madagascar and the Arabian Peninsula, with a lot of dispersal, vicariance, and species turnover occurring around 10 million years ago and more recently. Euphorbia, on the other hand, has had a much longer history of dispersal, vicariance, and structural diversification, with the global distribution as reflected in the phylogeny at the top on the right-hand side of this figure, multi-continental. This figure is from a recent paper by Arnest et al., just published just a few months ago in 2021. And these authors have made links between climate, history, and growth form over the evolution, and are talking about how this wide range of structural types may be linked to different climatic types, with taller woody forms associated with more mesic tropical areas, and these more modular forms associated with arid ecosystems. This is the base in Africa. And so this paper's got a lot of interesting insights. I've recently just got hold of it, and it's well worth reading. I'm still working my way through it. But two predictions from this work are that environments with less water stress for euphorbs tend to harbor more diversity of their growth forms. And the absence of fire disturbance has the same outcome, allowing more complex architecture to evolve. So absence of severe drought and absence of fire appears to foster innovation, structural innovation in euphorbs, according to these authors. And despite the differences in the age of crown taxa, it's clear that there's been a lot of recent diversification in succulents. And here I've got two phylogenies, one for the Azoaceae, one for the Euphorbiaceae. A little bit indistinct here because we try to get so much information across. But here we see the African clades of Euphorbiaceae. This is 10 to 0 million years ago, and a lot of diversification happening in the late Miocene and even into the Pleistocene. It's even more marked for the Azoaceae, with particularly extreme diversification occurring in particular sets of Azoaceae as little as a few million years ago. So certainly in the Pleistocene here. Something seems to have changed in the world that favored the spread and success of the succulent growth form more recently. We're starting to develop a better idea of what that might have been. And it's related to two important things, climate and CO2, both of which affect the performance of succulent plants. In the graph on the left, this Bierling and Royer paper, which shows how atmospheric CO2 is likely dropped from levels of about 1,000 parts per million, more than twice what they are today, to levels of prior to the industrial era, around 300 parts per million over the course of several tens of millions of years. And CO2 dropped below this critical level of 500 ppm around 20 million years ago. So this is when succulents start to radiate a little bit, and then they really radiate like mad here in the late Miocene. At the same time, the Earth's climate has cooled, the ice caps have formed, and driving further cooling and drying. And if you go and look at what that did to, if you look at the end of the Pleistocene and the periods of glacial times, the most recent of which occurred about 18,000 years ago, when the world was about 5 degrees cooler and the northern hemisphere had substantial coverage of ice. This is what Central Africa looked like. You can see that tropical forest was limited to these rather small patches, and the savanna and grassland biomes were in the ascendancy. And one wonders what happened to Thicket at these times. This seems to have been driven both by dryness and by low CO2. Very different time. We suffer a little bit from the hubris of believing that our present day climate conditions are normal in some way. But once you start studying paleoclimate, you quickly have this notion dispelled. It's sometimes a bit of an uncomfortable feeling, but once you get over it, I think that many patterns can start to make more sense, these ghosts of climates past, and we should be alive to them. How would succulents have responded to these changes? One of the most powerful ways we believe is the evolution not only of water storage capacity, but of a novel and derived photosynthetic behavior, so-called CAM, or Crassulacean acid metabolism. Understanding CAM is vital to understanding how many succulent plants behave. And I want to give you a quick physiology outline of how this works. And I really hope I'm not turning people off who don't want to be specialists in this, but I'll try to keep it as simple as possible. And it is quite interesting to understand this. But the ancestral form of photosynthesis looks something like this. This is a very basic schema here with the interior of the leaf represented here. This is a stomatum, the little holes in the surface of leaves that regulate water and CO2 flux out of the leaf into the atmosphere. And how C3 photosynthesis works is that light is harvested by chlorophyll, and it's used to generate chemical energy that drives the fixation of CO2 using this enzyme, ribisco. Ribulose 1,6-bisphosphate carboxylase oxygenase, for those who want to know. And this drives the Calvin cycle and produces these 3-chain carbon molecules, tricarboxylic acids, which is where we get the C3 moniker here for C3 photosynthesis. And the disadvantage of this kind of photosynthesis is that you have to open your stomata during the day when there's light, because that's when this whole engine runs. So the disadvantage is that during the day it's hot, can be dry, and in order to take up CO2, you can then lose a lot of water vapor. And that can be a real problem for plants that in dry environments. Another disadvantage is that this enzyme is not particularly efficient, and at low levels of CO2 particularly, it starts to fix oxygen and not CO2, and that's a wasteful cycle. So this kind of photosynthesis becomes very inefficient in dry, low CO2 environments. In other words, the very environments that have started to become much more prevalent as CO2 levels dropped. But what do cam succulents do? Well, they do something a little bit more elaborate, and it's not too difficult. Don't be put off by the boxes and arrows here. It's quite easy to understand, actually. What cam plants do is they split their carbon fixation between the day and the night time. They're quite an amazing process here. So what they do is they open this stomata at night when it's dark, and they fix CO2 using the enzyme PEP carboxylase, which fixes the CO2 into 4 carbon acids, these malic acids. And they get stored like a battery in the vacuole of the cell. And so that, you know, the plants are fixing CO2 at night when it's cool and the humidity is higher, and so they are much more water use efficient by doing this. And then during the day, when the sun comes out, they close their stomata, they seal off the leaf, and then they recycle all the stored carbon that's been stored in this vacuolar battery here. And that acid is decarboxylated and then goes into what is just a typical C3 cycle here. There's the original photosynthetic pathway operating as it always did. But the disadvantage here is that there's a limit on how productive the plant can be, because it's set by the storage capacity. So while you're gaining a lot in terms of water use efficiency, you're losing in the fact that you aren't as flexible. You can't respond when conditions are really good and be very productive. Now, in South African succulents, as always, with South Africa, there's a little wrinkle to the story. And we now know that South African succulents have found ways to get around this limitation. And our plants, many of our succulent plant species are able to switch between C3 and CAM. They can use both and mix and match. Some quite flexibly, actually, even from week to week, from day to day, they're able to switch C3 and CAM on and off, it appears. The way we can tell this is that the signal of the enzymes and the various processes in the leaf are captured in the stable carbon isotope ratio. It's a ratio of 13 and 12 carbon atoms. And I won't go into the detail, but basically C3 plants have a delta 13C of minus 23 and lower, and CAM plants will tend towards minus 10, somewhere around here. So this is a CAM C3 axis here, and we can see that many South African succulent plants are arrayed along this axis. This is data from Mooney et al. 1977. It's a study that's well worth updating by now. You can see Portulacaria afra here sitting right in the middle. A very flexible photosynthetic rate is indicated, can flip to C3 or CAM, and it's got this mixed signal as a result. But we've got many species that appear to be moving up and down this axis, and thus responding quite flexibly. So this is a really interesting ability to respond to rainfall variability, climate variability, and is that one of the tricks behind the incredible diversity of succulent plants in this part of the world? One thing that's a little bit counterintuitive about our succulents, given all this flexibility, is that they may not be particularly drought tolerant. And this is to be found especially in the Azoaceae, the mesems. And here I've got a sequence of photographs of an gyroderma in the Crassulacean taken by a citizen scientist who has been working with our lab. And we were monitoring populations through the drought of 2017 and into 2018. And we were able to capture, this is photographs taken by Hendrik van Zyl, a retired lawyer at Nieuwoudtville. who really diligently took a lot of photographs over many months. And we were able to see the course of drought in this particular individual here. And we can see they're still looking good in November 2017, but come December and then into January, we see the plant really taking strain, and then finally it succumbs at the end of January 2018. And we had mortalities of in excess of 40–50% in this population. So regular rainfall seems to be an important element, at least for this group and maybe others. Not too dry. To sum up, we can say that succulent plants have diversified in Pleistocene times. They have advantages conferred on them through their photosynthetic activity and their succulence that allows them to take advantage of low CO2 and dry conditions, not too dry. And by avoiding expensive construction materials like lignin and woody structures, they save on carbon as well. And they use water as a construction material. They use the pressure of water to construct their stems and their leaves and maintain their photosynthetic surface as well. So it's quite an interesting growth form in that sense. Lots of efficiencies built in, but with a few disadvantages as well. Now, how does this all relate to the thicket biome? And as I did in my talk of 2 years ago at the forum, I want to refer to the plant lifeform approach used by Rutherford and Westphal to define the biomes of South Africa. And in this slide, you see the original biomes. And I just want to remind everyone of how they were defined, because despite the nomenclature used for the biomes, succulence is not a defining feature of any single biome. The diagram that Rutherford and Westphal developed was this very simple one. It's actually deceptively simple. It required a lot of work, and to condense it and distill it in this form, I think is particularly brilliant. But what they did was to say that biomes could be defined by Brownkeer life forms, very large sets of life forms, trees, grasses, shrubs, and annuals. And so if you did this over very large areas, you could say that desert areas were defined by dominance by annuals, therophytes, grassland by grasses, the forest by trees, obviously, and then savannas by a mix of trees and grasses, the Namakuru by a mix of shrubs and grasses, Namakuru, it's a co-dominance of shrubs and grasses, and then fynbos, shrubs, grasses, and trees, the grasses being restioids. The succulent Karoo is defined by dominance of shrubs, not succulents. That's be careful with that one. A lot of people don't know that, forgotten it. And then there's this missing biome here that Rutherford and Westphal didn't define. So succulence is not a feature of any biome definition. And we've started to, I suggested that maybe this is the thicket biome. This is a biome that Rutherford and Westphal failed to identify because they just didn't have the data, co-dominated by trees and shrubs and shrubs and trees. And that's I think it's an idea worth dusting off and revisiting. What Rutherford and Westphal did that was really useful, and it's hidden in the pages of this report, this Memoirs of the Botanical Survey of South Africa, is to plot sites representing the different biomes on these two axes, a summer aridity index axis and a winter rainfall percent axis. So this is low winter rainfall, high winter rainfall, or high proportion. This is a proportional measure. And then the SAI represents an index relating to the amount of rain in the 4 hottest months of the year. And where you have an index of approaching 7 to 8, you've got very dry conditions in summer, and these are wet conditions in summer, right? So wet summers, dry summers, and proportional winter rainfall. And what they did was to define these growth form exclusion lines, as they call them, for shrubs, grasses, trees. And essentially how it works is that you don't find shrubs on the right-hand side of this shrub exclusion line. So shrubs are limited to that side. You don't find grasses on the right-hand side of this line, right? So the succulent karoo is dominated by shrubs, because shrubs can occur, but grasses are excluded. And the nama karoo, a co-dominance of shrubs and grasses, because trees are excluded on this line here. And you've got a little patch of arid grassland here where shrubs are excluded, but grasses do occur. And savannas and grasslands are found here. Fynbos is up here. You can see that there's an aridity gradient running from left to right here, wetter to drier. Forest is found here in these aseasonal environments. So what I did was to simplify this and plot the axes in a much simpler way. It's the same pattern. I've just hand drawn these exclusion lines for shrubs, trees, grasses, and put in the biomes. And what's quite fascinating is where all the biomes come together here in the middle of this area. This little node here, this nexus area. It's an area where there's aseasonality, and where there's it's not wet, it's not too dry. And you can define the fire-driven biomes something like this, where you have enough seasonality to have a dry enough period of time to support a fire, and enough fuel, because it's wetter on this side, so you've got more fuel, ability to build fuel, and you get these are the fire-driven biomes. This is the fire world, disturbance world here. And likewise, you can plot in the drought-driven biomes here, with the severe drought down here, where summers are dry and there's no winter rain, and then the slightly less severe droughts in the succulent karoo biome, where there's dry summers but wetter winters. And in the middle, you have an absence of all these stresses. So you can plot an aseasonal climate by filling in here, say, from 60 to 40% winter rainfall, pulling it across, and you're simply defining an aridity gradient here from wet, aseasonal, to dry, aseasonal. And as Julia Glende pointed out in the WhatsApp chat after one of the talks earlier this year, the climate of the thicket seems to fall into this little sweet spot here. And that's rather fascinating. It's a sweet spot where both fire and drought are absent to low frequency. And at this time, I don't want to go too far, but I just want to say that, ask the question, is this part of the secret of the diversification of succulents and maybe other growth forms in this biome? It's a locus in the summer aridity index and winter rainfall space, which was not highlighted by Rutherford and Westphal. This remained hidden in the somewhat forgotten pages of an old Memoirs Botanical Survey publication for decades. Is it time to pull this out, dust it back off again, and start asking some questions about it? And that's really where I want to leave it. I'll leave you with a set of questions that I developed for the talk a couple of years ago, and I won't read them out again. I can leave them up to you to read. And all I want to say is I hope you've gained something from the talk and are in for asking me some questions and getting a discussion going. I've really enjoyed putting it together and interacting with this group. And thanks to Alistair and Kath for asking me to do the talk.